Okay, so now is 1:30, so we will start our class and just open INU Smart Campus app to check your attendance. Just enter 9891 and also open your cam. Opened all of your cam. Okay, I will check it. 문경록 수업 안 들어갔는데?
네, 교수님 저 있습니다.
왜 출석 체크해? 아, 체크했구나. 오케이.
아, 네.
안다연? 없네. 정고연? 오늘 또 넣네. 지난번에 뭐 출석 인증해 줬는데 못했다고 하길래. 황선민? 천준영? 박지효? 그 에너지 화학공학과 이정길, 김지연, 최지연 잘 듣고 있지?
네.
오케이. 3명 다 오늘 다 들었네? 오케이. 오케이, so 나노바이오 신진현?
네, 교수님.
수업 안 들었다가 들었네? 어제 안 들어왔지? 아, 그 지난 시간 안 들어왔지?
네.
잘 따라가고 있어?
네, 잘 듣고 있습니다.
Okay. Hold on seconds. So we will start our class. Okay. So if you see here, you have to see the announcement. Okay. First, I announced the final term exam. So next week, okay, December 9th, okay, from 1:30 to unlimited. Okay, I think you guys, all of them can solve the final term exam within 1 hour. However, our exam time is unlimited, okay? And last time I also explained some important procedure. Okay. And also, I also explained the problem number and the point. Okay, total point is 100 points. Homework is 50 points, and final term exam is 50 points, is like this. Okay, so today we will cover from the chapter 8, middle of chapter 8, and we will cover chapter 9 and chapter 11, okay? And I will check our homework submission status. Everybody already submitted all of the homework. Okay, it's good. Okay, now we will go to the chapter 8 again. So do you have any other question in chapter 8 course or the homework? Do you have any other question? Okay, so let me go to the second subject, translation. So translation means that conversion of genetic information into protein. So there is a genetic code. This is the rules to translate genetic information into proteins. So we learned about the leading frame. Okay, so in our homework, we cover that leading frame. For example, chapter number 1 was the mention about the leading frame. Okay, and also codon degeneracy. In genetic code, there is 2 rules. First one is codon degeneracy. Second one is nearly universal. Okay. And also we will go to the transfer RNAs. Okay, so in the amino acid tRNA thin days, there are 2 classes, class 1 and class 2. The main difference is that class 1 thin days attach amino acid to 2' OH in the ribose. They have 2' and 3'. Okay, 2' OH of the tRNA. This one is class 2 is 3' OH of the tRNA. This is the main difference. Okay, and also we cover the ribosom and also cover the translation initiation and the elongation step. So elongation step is very important to make polypeptide. Okay, and also the thing is that new amino acid tRNA that is complementary to the codon bind to the A site. At that peptidal transferase, okay, peptidal transferase is happen. Okay. So finally, and also there is the translational termination. So RF1 is RF2 is involved in translation termination. Okay. And also, okay, we can go to some antibiotics that inhibit protein synthesis. Okay. So this is the amino acid tRNA. Okay. So they looks like this. So they have adenosine and the ribose. Okay. They have a phosphate. After they have tRNA is like this. Okay. So they have 2' OH. And this is 3' OH. For example, in this kind of the tRNA, they have the OH and they have the amino group is amino acid group like this. However, this is the puromycin structure. So puromycin structure is nearly similar to this is tRNA and this is the 3' end. So they are similar to 3' end of amino acid tRNA. That's why this puromycin can enter the A site and block the peptide bond formation. So they block the peptide bond formation. So it means that they will cause premature polypeptide chain release. Okay, they will cause premature polypeptide chain release. So in our homework, we cover this one. Important. In our homework, we cover that. So puromycin bind to the A site and prevent further chain elongation. Explain how puromycin acts to induce this effect. Because the puromycin is resemble 3' end of tRNA, so it enters the A site. After that, it blocks the peptidal transferase, so it will cause premature chain release. This is answer, okay? Important, okay? Another one is that, so this is the catalytic mechanism at a large subunit of ribosome. So in the large subunit of the ribosome, so they have the adenine monophosphate. So this is adenosine. So they have adenine monophosphate. So in the enzyme site of the large subunit ribosome, they have the adenine monophosphate. Okay, this is adenosine. Okay, this N we call it as N3, and this N we call it as N1. Okay, so usually this is the how peptide transport is happen, okay, at large subunit of ribosome. So initially, the N3, so this N3 received the proton from NH2 group of the amino acid tRNA. At that, nitrogen in the amino acid tRNA attack carbonyl group of the tRNA with the polypeptide. Okay, so they will make this kind of intermediate status. This one we call it as tetrahedral intermediate. Okay, this is we call it as tetrahedral intermediate. Okay, so after that, this is the remaining procedure is like this. After that, so polypeptide chain is made like this. Okay. Okay. So important thing is that Yaros inhibitor, another antibiotics. The full name of Yaros inhibitor is that this is puromycin. The Yaros inhibitor this is also puromycin, but they have another group, CCDA phosphate. So they have the puromycin structure here, and they have a CCDA phosphate group. So in addition to the puromycin group, they have a CCDA phosphate group. So actually so this one looks like the amino 3' end of the amino acid tRNA. Okay, and also this one is also looks like 3' end of the amino acid tRNA, and also this one around this region is nearly similar to the tetrahedral intermediate. So Yaros inhibitor function as comparative inhibitor of the tetrahedral intermediate. So it means that Yaros inhibitor inhibits peptide bond formation in the tetrahedral intermediate. Okay, so they inhibit they inhibit the peptide bond formation. Okay. So we also cover that in our homework. We also cover that Yaros inhibitor. Okay. So at that time, the question was draw the normal assumed tetrahedral intermediate. So we can write down here, and also the highlighting the tetrahedral carbon center. So this is the tetrahedral carbon center. Okay, so in actual exam, you don't have to memorize the all of structure. So I will give you the structure of the subunit in the important important component in the large subunit ribosome. And also I will give you the one tRNA, I will give you another tRNA structure like this. Okay, so you know that some important procedure here and there like this. Okay. Any other question in the Yaros inhibitor? So any other question in the puromycin? Okay, it's very important. Okay. And also, finally, we cover the protein sorting and targeting, because this secretive peptide. So secretive peptide is important for translocated protein from the ER to Golgi. After that, they will secrete the protein is outside. And this is the N-terminal sequence of secretory pre-proteins. So they have the secretory signal is like this. So they have arginine or lysine, arginine. They are positively charged. After that, they have the hydrophobic region is like this. Finally, this is the cleavage site like this. Okay. So to make the secretory protein, we usually add this kind of sequence like this. In the company, we want to make so in the company Celltrion or Samsung Biologics, they want to make the antibody. Okay, so to make the antibody, they always add this signal in front of the antibody. So it means that antibody can be secreted from ER to Golgi. That antibody can be secreted. Okay, at that the signal peptide can be cleaved like this. Okay, so at that in the company we always collect the media, not cells. At that in the media there are lots of the protein, lots of the antibody. After we always do sorting in and sorting out. We can precipitate the protein by using sorting in and sorting out method. Okay, this is very important. And also we cover the SRP. This is the signal recognition particle. Okay, they have this kind of the M domain. M domain is a long hydrophobic group is like this. Usually they recognize the signal peptide for secretion. Okay, like this. Okay, so and also we cover this concept at the final question. We cover this one. Okay. So this is the M domain. They have the long hydrophobic group is like this. Okay. This is signal peptide binding site. And also another targeting signal peptides for targeting protein mitochondria. Okay, so now we will cover signal peptide for targeting protein to mitochondria. They have the basic residue, they have highly percentage of the protein which has the basic residue, arginine or lysine. Okay, after that, this one is recognized by Tom that they were transported into the inner membrane. Okay, after that, usually the signal peptide is cleaved by another protein is like this. Okay, and we cover this one in our homework. So, so signal peptide for mitochondrial targeting protein is lysine or arginine. They have a highly residue, the percentage of the lysine or arginine. important. Okay. Okay, important. Okay. Another one is the signal peptide protein targeting to the nucleus. So this is the nuclear localization signal. So they are highly preference. They have a high amount of lysine, arginine, or proline. Okay, mitochondria targeting sequence is lysine, highly percentage of lysine, arginine, but in addition to lysine and arginine, so they have the highly percentage of proline like this. So usually the nuclear import proteins, so they bind to the NLS of the NLS, and after the import protein is inside of the nucleus, is like this. However, NLS sequence is not removed even after protein is transported inside the nucleus, because they always reused. Okay. Okay, so this is the sometimes some metazoan, they did open mitosis. So if they don't have the NLS sequence, it means that they will lose that protein after they make the nucleus like this. That's why usually the NLS in the cargo protein is not removed even after cargo protein is transported inside the nucleus. So we also cover this concept in the homework. Okay. So okay, we okay. So unlike the mitochondrial signal sequence, NLS is not removed. What is the reason? Because the main reason is that the so NLS can be reused many times in the normal life of the proteins. Okay. Okay, so we covered the chapter 8. Now we will go to the chapter 9. Okay. Hold on a second. I need water. Okay. 1 minute break. Okay. Okay. Okay, let me go to the chapter 9. So do you have any other question in chapter 9? So any other question? Okay. Any other question? Okay. Okay, now we will go to the chapter 9 chapter 9. So chapter 9 is, we call it as protein expression, purification, and characterization. So in the Celtrion and Samsung Biologics, they always do this step. It's very important step. Okay. So if you go to the company, you will do part of this work, the protein expression or purification or some characterization. Okay. Chapter 9 is composed of the recombinant DNA technology and protein expression and harvesting and purification. Okay, so this is the pharmaceutical manufacturing process from cell culture, harvesting, purification, and manufacturing. Their step is very simple, very simple. Okay. Okay. And also, so now I will cover the first step, cell line development. So when I was in the bioepis, Samsung bioepis, I was the head of cell line department, cell line development department. Okay, I was head. There are several head. There are several head is there. So I was head is there. Okay, so if you, for example, if you want to go to the cell line development part, so you need at least a master degree, because after you finish undergraduate, so you always go to the harvesting step, or sometimes you go to the purification step, or usually the people who finished undergraduate always go to the QC or QA. So after we purified protein, before we manufacturing the proteins, we always do QA or QC. So you will go to this part. But if you finish it master degree, so you always go to the cell line development part. Okay. So this is composed of this step like this. So I will briefly explain that part. Okay, so vector is most important part in the cell line development. Okay. So who is developing the next generation vector platform? So I am developing the next generation vector platform to produce more proteins. So my research focus is to make the vector. So if you are interested in this step, and actually this step is the most important part in the biopharmaceuticals. Okay, so our lab is the studying about the vector. Okay. So this is the vector structure. Is mainly composed of the origin of replication, and they have the antibiotics resistance gene. And finally, they have the multiple cloning sites like this. Okay. So we always cloned our gene of interest in the multiple cloning sites because they have the multiple restriction enzyme cutting sites is like this. Okay. And also this is the detailed vector structure. So to make the insulin, we always use the inducible promoter. So this is the basic principle. Okay, and also they need a shine diagonal sequence to make the proteins. So this is start codon, is composed of AUG. So it encodes the methionine. Okay, so before the methionine, we need shine diagonal sequence. So this is composed of AGG, AGGU. So when I was in the company, I asked some researcher to design the vector for expressing some proteins, but he did not add the shine diagonal sequence. It means that protein is not expressed. Okay, so I asked him, you missed this very important step. Okay, so shine diagonal sequence is needed. Okay, also another one is like this. Okay, and also the most important part in the cell line development is cloning. It's process of constructing new DNA molecule. Okay, so we use the restriction enzyme, and after we ligate, it's like this. So who is the cloning king in the world? So I am the cloning king of in the world, I think. So I made 1,000, maybe I made 1,000 of construct, maybe. So nowadays the number one cloning king in the world is the person in my lab, our master degree student. He whenever I give him some subject, he always make he always make the construct just within 1 week. Okay, so I call him as you are cloning king. Nowadays you are the cloning king. Okay, cloning is like this. Okay. Now we will go to the second part, harvesting. So harvesting is composed of so you usually after you finished undergraduate, you will go to if you go to the cell trion, maybe you will go to this part. So you always harvest the cell. Okay, so this is composed of cell wall disruption. For example, gram-positive bacteria. So we did the mechanical disruption, and also gram-negative bacteria, we do the enzyme disruption method. After that, we always do centrifugation. So during the lecture, I explained the detailed procedure. So finally, we get this one, supernatant. So supernatant, they have the last of the protein complexes. Okay. But, okay, last of protein complexes. So after that, so this is the main principle of centrifugation. So RCF is the relative centrifugal force. Okay, they are composed of omega 2 multiplies omega is the angular velocity radians per seconds. So this is the equation is like this. Okay, R means that rotor diameter, and G is like this. Okay, so this is question. What is the average relative? What is the RCF in 10 centimeter rotor spun at 10,000 RPM? So we can calculate it based on this equation. It's like this. Okay. So finally, this RPM can be changed to the RCF like this. Okay. So we in the company we always used this equation to change it RPM to RCF. Okay. You have to understand this concept. Okay, in our homework, we cover this one because this is very important concept in the company. Okay, because you learned the protein engineering course, so it means that you have to you can calculate this equation like this. Okay, very important. Okay, so you need a calculator during the final term exam. Okay. You need a calculator, okay? So, and third part is purification. So if you finish the undergraduate, maybe some of them will go to the purification part, okay? The important thing is that the concept is ionic strength. So ionic strength is like this. This is like this. M is that concentration of ionic spaces, and Z is charge of all ionic spaces. At that, so this is equation is like this. Okay, how about let me go to the one example. The ionic strength of the 0.75 mole solution of MgCl2. So what is the ionic strength? Okay, Mg2 plus concentration will be the 0.75 mole, and charge of Mg2 plus will be the +2, isn't it? Okay. How about Cl minus? Cl minus, they have 2. So the concentration is 2 multiply 0.75. Charge is -1. So we can calculate it like this. Okay. Very important. That's why it was shown in our homework. That's why it was shown in the homework. Okay. Important. Okay. You need a calculator. Okay, now we will go to the sorting in and sorting out step. Okay. So after centrifugation, you got the supernatant. Supernatant containing the protein complexes. Okay. In the case of the insulin, to make the insulin, we did not add a secretory peptide in front of the insulin. That's why we have to disrupt the cell, and after we have to centrifugation. However, in the kind of some antibody, so we always add secretory signal peptides for secretion like this. So it means that we only can collect the supernatant. Okay. So in this case, we just collected supernatant, the media, or in the case of the insulin, after we did centrifugation, we can collect supernatant. Both cases take the media or the supernatant containing the proteins. To precipitate protein, we always do sorting in and the sorting out method. Okay, so I explained this procedure during the lecture very detailedly. Okay, sorting in and sorting out is like this. During the sorting in, so it will increase protein solubility. And during sorting out, okay, so it allows the protein to aggregate and precipitate it like this, okay? And also we learned about cosmotropes, and we learned about chaeotropes, is like this, okay? So this is the one example of the cation, one example of one example of the cosmotropes and chaeotropes. Okay, for example, this one is the this one is the most good cosmotropes ion, and this one is the chaeotropes ion is like this. So guanidinium is chaeotropes ion. Okay, and also thiocyanate is also chaeotropes ion, and also this one is also cosmotropes ion. The reason why we always in the company use ammonium sulfate in protein purification. So we always use the ammonium sulfate to precipitate the protein. So we are doing the sorting in and sorting out. This step we always use ammonium sulfate. So there is two reasons. So based on this graph, you will see that ammonium sulfate is very soluble in the water. It means that it can reach. This is the it can reach at highest concentration. It means that it can reach in the highest concentration. It means that it can result in complete precipitation of proteins. It can, so if there are, for example, there are 100 of protein is in the supernatant. But if we use the ammonium sulfate, we can precipitate 100 protein. But if you use another one to precipitate the proteins, so for example, if we started at 100, maybe you will get only 50, like this. Okay. So another one is that so we can so they are the cosmotropes. Okay, we can precipitate in native structures like this. Another one is the purification. Is the after precipitation, we always use the chromatography. One is ion exchange chromatography, and the other one is the size exclusion chromatography. Is like this. Okay, let me go to the ion exchange chromatography. Okay. So in the company, I explained this concept, but no one knows about this concept very well. So now, but during the protein engineering course, we cover this concept. Maybe you guys already understand this concept, I think. Okay. There are two kinds. Anionic exchange chromatography means that molecule of interest is anionic. Okay, so molecule of interest is this anionic, negatively charged. And this chromatography, they have positive charged resin. They are composed of positively charged resin with it. How about the cation exchange chromatography? They are opposite, totally opposite. Okay. Okay, so this is the one example of anionic exchange chromatography. So they have a positively charged resin is like this. There are two kinds of the QAE and DEAE. Okay, so QAE is strongly bind to the negatively charged protein, and DEAE is a weakly bind to the negatively charged proteins. So also in the company, we always use the cationic exchange chromatography. So we are interested in positively charged protein. So at that time in the resin, we are using the sulfur propyl or carboxyl methyl. So their resin is negatively charged like this. Okay, so in the company we always use the cationic exchange chromatography. So we have the resin. So the resin is negatively charged. So we always use SP or CM. So in the company, I explained this one, but no one knows about the structure like this. No one knows why SP is strongly bind to the positively charged protein. But we learned their structure is like this. So important. Okay, so because we learned this course is protein engineering course. Okay, so now we learned ion exchange chromatography, especially cationic exchange chromatography. So we always use this SP or CN. Okay, SP can strongly bind to the positively charged protein, and CM is weakly bind to the positively charged protein. Their structure is like this. So in the homework, we say that which one is more strongly bind, this one, okay? And also write down the name and their structure of two highly used resin. One is SP and one is carboxyl methyl, like this. Okay, you have to memorize, okay? Okay. Now this is size explosion. So based on this graph, we can estimate the protein, we can estimate the protein size Okay, so this is total elution volume, and this is the logarithm of molecular mass. They shows the linear relationship. And also, for example, very small proteins, we always add PEG. The reason is we just want to make this one into here. So we can estimate the apt. Any question? Okay, so we can estimate the apt maybe 10 milliliter of running buffer has run out in the column. Maybe we can get this molecules. We can estimate it. Okay, so this is so we cover this one in the homework. Okay, and also another one is affinity chromatography. And also in our company, we always made antibody, therapeutic antibody. At that time, we used affinity chromatography. So we always use the protein A resin or protein G resin because they can bind to the Fc region of the antibody. It's like this. Okay. And the price of resin is very expensive. For example, 100 gram of the protein A resin is it is about it's very expensive. I cannot remember exact number, but very expensive. Okay. Okay. So if you want to make the more money, so you can make the another protein, resin and protein A or protein G, or you can make the protein X like this. So maybe you will become the billionaire. Okay. Okay, but the region protein A or G can bind to the Fc region of the antibody. Okay. And also there is another one is nickel affinity chromatography, because so polyhistidine can bind to the nickel. Okay, so whenever we make the protein, so we always add at the end, we always add the histidine tag. So it means that we can purify the proteins very easily. So if you want to learn this procedure, come to my lab. We always do this kind of experiment. Okay. Okay. Okay, so we finished chapter 9. Chapter 9 is very important step in the company. Okay. Okay, so we chapter 9 we learned we have to know the important, we have to know this is the we have to understand centrifugation. And also this one chapter number 2 was number 2 is summarized. So if you understand all of the procedure, you can solve number 2. So in number 2 there is no answer. This is one kind of answer. Okay, so you can propose another answer. Okay. Okay. This is my answer, and your another answer. If your answer is logically right, so I will give you full score. Okay. So it means that if you have this kind of question, to solve this kind of question, you have to understand all of the procedure in the protein purification. Okay. Very important. Size exclusion, or I used first I used size exclusion. Second, I used nickel affinity. Finally, I used anion exchange chromatography. It's like this. Okay. Okay. So you will see that. Okay. We will cover important procedure. This one, Ionic Strength. Okay. And finally, during purification, SP and CM, we covered it. Okay, do you have any other question? Okay, so now we go to the chapter 11. Chapter 11 is the name is the protein folding in vivo and in vitro. Okay, do you have any other question in chapter 11? 김영우 잘 이해하고 있어?
네, 잘 이해하고 있습니다.
이제 김영우 나이가 많던데
네.
사업하고 있다 했나? 아, 김영우가 아니야?
네?
요 수업 듣고 있는 애들 중에 사업하고 있는 애들 있던데 보니까.
아,
오케이. 안성준 잘 듣고 있어?
네.
잘 따라가고 있어?
네.
오케이. 열심히 해야 돼, 알겠지? 열심히 해야지 A 플러스 받을 수 있어, 알겠지?
네.
Okay, you still have a chance to get A plus, okay? Okay. Thibaut, are you okay? Everything is okay?
Yeah, everything is okay.
Okay, so because the chapter, we cover chapter 9 is also very difficult. Okay, but I explained very important step. Okay, in chapter 9 we have chapter 9 we have 4 questions. Okay. Now we will go to the chapter 11. Okay. Protein folding in vivo and vitro. So this is this kinetics we always use in the company. We always use this one. Okay, so now we will cover factors determining protein fold, and also we will explain the amide exchange and measurement of protein folding. Finally, we will cover kinetic barrier to refolding. Okay, first one is the factors determining the protein fold. Okay, so this one is denaturing, means that process in which proteins lose their shape and function. So this is native structure, and this is denatured structure. And folding means that process by which linear proteins form their own folded structures like this. Okay, and also the first factor is denaturant. Okay, we covered this one. We covered in chapter 9. Okay, so at that time we covered cosmotropes and chaeotropes. It's like this. And also, so now also, now we also denaturant. We also have another denaturant, guanidinium. Denaturant is belong to the chaeotropes ion. So guanidinium. This is guanidinium structure. Another one is urea. So urea is nearly similar structure to guanidinium. So okay, so in the company we use the urea better than the guanidinium. Okay, but they are nearly similar structures like this. Okay, and also we cover second factor is hydrophobic effect. Okay, so hydrophobic effect induces barrier of hydrophobic residue in the center of the complex is like this. Okay, this is water. Another one is accessible space area. Okay, so accessible space area is increased as a protein is denatured, or the concentration of denaturant when this is increased as the concentration of denaturant increased, and also there is another one is like this. Okay, now we will go to the very important procedure, and also I announced some typo. Okay, so I already announced the type correction. Okay, so the first one is we have to know the pre-energy changes delta G between the unfolded and folded. And the delta G is, in this case, delta G means that delta G is the free energy of G folded status minus free energy of unfolded status. Usually this value is always negative. It's like this. So it means that reaction from the unfolded to folded status goes spontaneously. This is thermodynamically favorable processes. Okay, this delta G, but we also use same symbol, but another terminology we have to know is that conformational stability. We also use the delta G. So this delta G is that free energy at G unfolded minus free energy and G folded is like this. It means that this delta G value is always positive. Okay, they use the same symbol, but they are totally different. Okay. Okay, so so this is the conformational stability. Okay, so unfolded can go to the folded. Okay, so this is the energy favorable processes, and sometimes folded protein can go to the unfolded the status in the absence of denaturant. So this is, we call it K1, and this is K2. So K equilibrium constant, KEQ means that K1 divided by K2. So we have the, so this value can be, we can calculate it by the concentration of folded divided by concentration of unfolded proteins. And also this conformational stability. So this is conformational stability can be calculated by using this equation. This R means that universal gas constant. The R value is like this. T means the Kelvin temperature. Kelvin temperature means that 273 plus the Celsius temperature. Finally, this is log, but this is a natural log and KEQ. So it means that the cation was at the midpoint of the unfolding transition between the folded and the denatured status. What is the KEQ? KEQ means that concentration of the folded divided by concentration of unfolded means that KEQ will be the 1 is like this, okay? And also this is very important. Okay, this is very important. In our homework, we also solved this one. It's okay. The rate of unfolding in the absence of denaturant means that K2 is 10. So while that the rate of refolding is like this, rate of refolding is K1 is value is like this. The process is two-state unfolded or folded. The best estimate of conformational stability at the room temperature. Okay, so based on this equation, we can calculate the post-calculated KEQ. At that calculated delta G, conformational stability is RT, and natural log KEQ, the concentrate the conformational stability is like this, 25 kilojoule per mole is like this. Okay, so this is, we have to know, okay, conformational, you have to know how to calculate conformational stability. Okay, okay, because we learned this equation we always used in the company. Okay, and also conformational stability in the presence of denaturant. So we cover this one in our homework number 2. Okay, homework number 2. So based on this one, we have to draw the graph is like this. Okay, now I will explain once again. So in the presence of the denaturant, the conformation, to calculate conformational stability, so we have to know delta GU H2O, and also we have to know MEQ. MEQ is binding constant between denaturant and protein, and D means that denaturant concentration. Okay, so the equation is like this. The equation is like this. Okay, so let me go to the let me go to the question homework. Okay, so we have the graph, we have the experimental value is like this. Observance 1 is we can estimate as all of protein is folded. Observance 0 is all of protein is unfolded. And also we can assume that MEQ is like this. And also experiment was done in room temperature. Okay, the equation is like this. So you don't have to memorize this equation. Okay. Okay, so we can draw the picture is like this. So to solve the problem, so we have to know, find the denaturant concentration when half of the protein is folded and half of the protein is unfolded. Okay, the concentration is about 1.5 mole. Okay, because at that time, delta GU, because this is a natural log KEQ, so KEQ will be the 1, means that natural log 1 means that it will be the delta GU will be the 0, is like this. So now we can calculate conformational stability in the absence of denaturants. So this is like this. So now we can calculate it like this. Okay, delta GU H2O will be like this. Okay. Okay, so we have to understand important procedure. We have to understand this one, and also we have to understand this one. Okay, you don't have to memorize the equation, but delta GU will be the RT natural log KEQ. Okay. Okay. So in the graph, we always have to know the half of, when the half of the protein is folded and half of the protein is unfolded, we have to know the concentration of denaturant. So, so in these cases, this is the 2, or this is more 3.5, something like that. Okay. Okay, very important. Very important. Okay. Okay. now we work another factor is temperature, and also in our homework we cover thermal stability curve. Okay, and this is the half of a protein is the folded and unfolded. So this is we call it as a transition midpoint TM. TM means that transition midpoint temperature. For example, in this case, transition midpoint temperature is about 70 something. This one is about 40 something. So it means that this one is more stable than 2. 2 is more stable than 3. 3 is more stable than 4. So we cover this one in our homework. So this is also differential scanning calorimeter. So in our lab, we used this method to study the protein. Okay, so DSD can measure the delta CP and TM. So based on this heat flow, we can calculate the TM. Okay. Okay, how about like this? So this is, we call this peak. So we can change it, this graph is like this. Okay, after we corrected the calorimetry profiles, we can make this kind of the graph is like this. This is TM, okay? And so this status is folded and this status unfolded. So we cover this one in our final homework. So we covered what is the TM. TM will be 60. And this is folded protein, and this is unfolded protein. Okay. So we always, in our research, in our lab, we always used this graph to predict TM. Okay. So if you want to measure the TM, just contact our lab. So I will show you the DST. Okay, and also finally we will cover amide exchange and measurement of protein folding. Okay, so this is also highly used method to measure protein folding. Okay, the principle is that the hydrogen in the amide group involves in the hydrogen bonding. This is the process is like this. Okay, there is difference between the proteome and the deuterium. Okay, deuterium, okay, they have the nitrogen. It's like this. So during the lecture, I explained the procedure. Okay, the important thing is that after we finished NMR, the higher concentration is deuterium. Okay, so you have to understand this concept. If they are the higher concentration of deuterium, indicate that protein has a higher folded structure. So if you have the lower concentration of D2, means that you have the higher frequency of unfolded protein. Okay, so this is amide exchange method can measure the degree of protein folding. So so today I did not explain the detailed procedure, but you have to know the concept of higher concentration of deuterium and the concept of lower concentration of deuterium. Okay. And finally, we cover kinetics barrier to refolding. So, so when the OP pathway is present, so this can be the kinetic barrier to refolding. Okay, if there is a X or this OP pathway is present, it will become the kinetic barrier between the unfolded and folded. It can become the kinetic barrier. The very famous example is that cis-trans isomerization of the peptide bond, especially in proline. Most of the cases is trans configuration, but sometimes they have cis configuration. So usually the cis configuration of the proline structure, they are kinetic barrier to refolding. Okay, so we also covered, this one was covered in our chapter 11 homework. Homework 1 is like this. Okay. Okay, so we covered the protein folding. So you have to understand important concept, conformational stability. Okay, this one. And also you have to know how to calculate this one. This one. And also you have to know this concept, conformation stability in the presence of denaturant. The very famous example we already solved in our homework 11. Okay, you have to know. Okay, we have to cover several important steps in the DSD. And also we covered some important concept in amide exchange, and we have to understand the final value. Okay, do you have any other question? Okay, so this is our course material, protein structure and function. I already uploaded this one, our course announcement. So if you have this book, you can see that we covered all of our. So today we finished chapter 11. Chapter 11 is located here. So we covered around here. We covered all of our books, okay? And we already solved all of important. So each chapter they have the problem. But you guys already solved all of the problem, okay? Okay. Do you have any other question? Okay, so 박나영, 시험 잘 볼 수 있겠어?
네.
시험 잘 볼 수 있겠어?
네.
오케이, 시험 잘 보면 되지 맞지? 오케이. Okay, so do you have any other question? Okay, so see you next Thursday. Okay? Okay, goodbye. Bye.
감사합니다.
고생하셨습니다.
감사합니다.
감사합니다.
감사합니다.